सेक्सनलाई म पुनः स्वागत गर्न चाहन्छु र यहाँलाई धेरै स्वागत छ।
Okay, so I'll try to keep this session a little interactive. So, sorry, I have in-semester exams coming, so I can't make it. So, very experienced chemist tonight. Then sirs tried, let's call a very talented chemist, working for Pfizer. That guy said, our drug is going for FDA review, so I can't make it. So very experienced chemist couldn't come, very talented chemist couldn't come. Then sir thought, let's invite a very handsome chemist, That's how I will invite him. So let's keep this little interactive, okay? Whatever questions you have, feel free to ask anytime, stop, okay? Drug development, right? So what is the role of a chemist? That's what I will highlight. And more than giving a lecture, I will try to only talk the things which I have worked or I have been seeing somebody work. So I'll try to relate it to my personal experience or things which I was trained. So to keep it more interesting. So that's what NIRI. It's a very Nepali-based organization. Nexus Institute of Research and Innovation. All the Nepalese scholars all around the globe are connected to it. So there are regular NIRI seminars. They also have some boot camp programs like bioinformatics boot camp. We are also thinking of starting a drug discovery boot camp. So most of you are welcome to join those classes. It will be online. That might start sometime soon. And just to say, like, if you have to talk about how drug discovery started, if you have to go back in timeline, like 1800s, 1900s, it was more like a serendipitous discovery, right? Suddenly Alexander Fleming found that one day a small mold, a leaf of a mold fell in his bacterial culture, and he saw that there was inhibition. And that's how he discovered penicillin, which is the drug. And then the rational drug design wasn't developed back then. It was just a serendipitous discovery. Either it was traditionally whatever was known. They knew what works from the experience, but nobody knew what was the active ingredient. Another example of that is cinchona bark, from which the malarial drug was extracted, right? So that's how was the drug discovery in the old days. And then it became so random, as you can see, like it's just you kind of mix it and see what happens, But then as the scientific method developed more and more, people started thinking, let's give it a firm footing, let's rationally design a drug rather than randomly screening thousands and millions of essays, let's do it rational drug design. So as it picked up, so chemistry started becoming a little more streamlined. This is what we call the rational drug design. Here you are thinking rationally, how you can particularly arrive at a molecule that will have a desired bioactivity, right? So that's what is called rational drug design. As you can see, there is a long pipeline there. But always all of that starts from the first process is like target identification and validation. So for every disease condition, there is always a target which you can target, right? So it could be a protein, or it could be any other enzyme, right? So if it is a bacterial antimicrobial drug, then it might target cell wall of the bacteria. It might inhibit cell wall synthesis, or it might rupture the, right? So those are those mechanisms of action. That's what you call mechanism of action, how the drug works, how the active pharmacological ingredient, API, right, or the active ingredient of the drug, how that works at the molecular level. That's where it starts. Now it's not a random thing. You look at basically you do a lot of in silico work, you do a lot of molecular modeling, and then you do a lot of docking studies. You find out certain pockets where certain molecules could bind. And then from there it starts. Once you come up with a molecule which tightly binds to any pocket of a disease site, then you try to synthesize that. That's where, so first comes in silico. You basically design it molecularly, you model it, then you synthesize it. That's where the synthetic organic chemistry comes very handy. So you have a molecular structure. All of you might have studied retrosynthesis we do, right? Disconnection approach, all of you are aware of that, right? So you have a target molecule, and then if it is really a bulky molecule, then how would you arrive at that? And you would like to look at loose connections where you can disconnect that, and then do a retrosynthesis analysis, and then from the small starting ingredients, how you can arrive at that, right? That's how basically large molecules are synthesized by synthetic organic chemists. So basically, you identify your target and you validate that. You have to validate that that's the target, and it doesn't have off-target effects. Those are so many things. I'll skip nitty-gritty details, but I'll try to give you an overview. And then basically, you have to screen it. After you know the molecule in silico, then you have to do a basic screening, like a prototype study, how does it work quickly to arrive. And then you also have a lot of things like toxicity studies and dissolving how like water soluble or lipophilic or hydrophilic it is, because those properties will become very important as you go further, right? So that those kind of things are quickly done. And then initially, if you come across any promising molecule, that's what is called hit. You hit something, right? So while you are screening it and something hit there, something looks promising. So that's just a hit there. So from there you do further optimizations. You might be aware of the structure and activity relationships, SAR, right? SAR studies. You have a basic skeletal molecule. If you are trying to bind into a certain pocket of a protein, and then you see that there is one let's say really hydrophobic pocket is not allowing it to, it's repelling it. Then what do? You would insert like OH hydroxyl or any hydrophilic group at the end, right? That kind of side chains. And then you again try to see if it fits there. So that's what is called structure and activity relationship. If you change the structure, how does it change the activity? And then you just go back and forth. That's how you optimize, right? That's what is called SAR study. So those kind of things are performed after you basically come up with a hit, and then it's also this whole process is called lead development. So after hit, if it further checks few more parameters, it becomes a lead compound. Lead compound is that something which has some potentials to be developed into drug, but of course it's a very long process, which I will say further as I go further. And then NCE is like basically new chemical entity, right? So and then finally it goes to drug. It's a very long process. Now the question is, what's the role of the chemist here, right? So traditionally, I was asking Bhanu sir feedback on what I can tell. Then sir told, we tend to basically segregate, like this is physical chemistry, this is organic chemistry, this is inorganic chemistry, or even this is physics, this is biology. But as you go further for real life applications, particularly for a medicinal chemist, you have to know every damn thing. There is no biology, chemistry, there is only science there. Okay? So you have to know the biology, you have to know the role of a chemist is basically an interdisciplinary role. Person to synthesize a molecule now. There are companies like Pfizer and Bayer, and like they are synthesizing like millions and millions of small molecules. They have libraries already screened. It's just like it's very unlikely that you are coming out just like de novo molecule. So what they have done, good thing is, of course, they don't share everything, but they have those databases. Whatever chemical entities they have synthesized or screened so far, and they have that kind of databases. And most of it is like basically a data mining also. So that's what the software comes in. So now a chemist also has to be a good software engineer. If not engineer, at least you should know some data science or how to use basic softwares or analyze data. That's another role a chemist has to take over, right? So basically there are all these lot of databases. People have synthesized the molecules and tagged them and made the libraries, and those information are available databases, and so that saves a lot of time. If you are working in something and which already somebody has worked, then it's a waste of time, right? So that's where it comes in. So that's the in silico aspect. So there is how many of you have taken medicinal chemistry as a course or drug design? Okay, but I will tell you something. There's a big guy called Lipinski. If you read medicinal chemistry, he's called Chris Lipinski. He was working for Pfizer. So I was talking about earlier about the databases, and then people have that access. So this guy had access to the database from Pfizer, like millions and millions of compounds. So he had the data, and he was a desk scientist. So he had the time, he had the data, and he had the laptop. So what he thought was, let me see what are those FDA approved drugs or the drugs which are out there in market. What are the common properties in those drugs? So he did a kind of a empirical statistical analysis. He took like thousands and thousands and thousands of molecules and just tried to see trends. What are their molecular weights, or what are the hydrogen basically donating groups they have on their side chains, or what is their basically lipophilicity or hydrophilicity, right? So he did this kind of empirical study and came up with this what's called Lipinski's rule of 5. Because you can also look at it in Google. It's a very whenever you come up with a molecule, you can try to check these parameters. It's not like there are exceptions, of course, for biologics or protein-based drugs or even RNA-based drugs. Don't comply to Lipinski's rule of 5 because they have more molecular weight than 500, right? So why he called Lipinski's rule of 5 is basically he came up with this, a very likely drug candidate should not have more than 5 hydrogen bond donors on the exposed side chains, and it should, like, this is not exactly 5, but he says 5 into 2, so it should not have like more than 10 hydrogen bond acceptors. And then the molecular weight should be less than 500, and then in the log P, basically it's a measure of how hydrophilic or hydrophobic a compound is. It should not be the C log P value should be greater than 5. So these are called Lipinski's rule of 5. So when I was at the University of Massachusetts, so he was a visiting lecturer there to teach us the drug design. So that's why I took a picture with him and said high five with Lipinski, because he has this rule of 5. So this is something, as I was telling earlier, so from the data, you have that enormous data, like using AI, people are coming with, like, you analyze, use the databases and then try to do empirical studies and come up with likely candidates. So you screen out a lot of, from like, let's say 1 million molecules. Just going through this process, you rule out like maybe 900,000 and you are left with 100,000, right? From there, you go ahead. You do cellular testing, biology, other studies. That's how you rule out. And then finally, whatever candidate checks all marks, that becomes like a drug candidate, right? So that's how the process goes on. So anyway, so that's the Lipinski's rule of 5. And as I was telling, the first step is data, looking at the databases and looking at in silico work. This is what is called in silico, right? So once you come up with a molecule that relatively binds to a disease site, then you synthesize that, right? That's where the synthetic organic chemist comes. So that's what I was saying, the docking studies. So these are the enzymes involved with inflammation. In fact, even COVID, when people used to get a lot of inflammation in the lung, right? They have the role to play there. So some of those drugs like dexamethasone and some other anti-inflammatory drugs do inhibit some of these enzymes, not exactly this. These are classes of, there are cyclooxygenase 1, 2, and there are also lipoxygenase. So these are basically class of those cytokines which are involved with inflammation in the body, right? So in this particular site, they basically the docking study, this was one of the research. As you are all aware of the curcumin, right? Yes. Our former Prime Minister, Sharma, was very famous for telling that curcumin can cure COVID, COVID. So he wasn't entirely wrong. I mean, but chemically, if you have to analyze further, the problem with curcumin is you see like it has this bulky rings here and then a lot of long chains of curcumin. It's just your body can't absorb it because it's very hydrophobic. That's why actually in our old days it was suggested you take turmeric with milk. Right? So milk has some fat, so it slightly better dissolves that way. So our tradition wasn't entirely wrong. They didn't know what they were doing, but they were doing right thing, And similarly, Oliva wasn't also entirely wrong. But if you have to look from a chemist angle, he is not a chemist, so we can pardon him that. Right? So anyway, so I was talking about the curcumin. So these days we know that this pharmacophore, whenever there is an active, you see a molecular structure like a skeleton, which binds to certain pocket. So you can stay fixed. It's like pillars of a house. You have the robust system, the skeleton. All you need to do is like a little bit fixing here and there, tweaking here and there. That's what is structure and activity relationship. So it's well known from ages that curcumin has some anti-inflammatory properties. So my work when I was in India, actually in India, Pakistan, and South Korea, a lot of natural product research happens, even in China and Japan also. So basically in India, particularly they were looking. So where I was doing my masters, I was working on this. So a group of basically computational chemists came up with this docking studies, and they figured out that more or less, if we can make a curcumin here, this curcumin, as you see, it has 2 ketones here, right? Yeah. Can you see that? Okay. So what is that famous reaction of molecule which has 2 ketones? It's very unstable. It just flips. What's that reaction? Anybody who answers will get a chocolate. What happens? Keto? Keto and not? So you get it. Awesome. So you know that it can basically your keto, if it picks up a hydrogen from the surrounding, it can go into in all form, right? Basically it becomes all in and then all over. So that's what it's called keto enol tautomerism. It's that what it does is basically it makes it very unstable, you know. Apart from its poor bio distribution, that's one more problem. So some of our in our research we were trying to see if we can strip off one of the ketone here. If it is not a diketone anymore, then it will not flip and do the ketoenol tautomerism, right? So that's why basically we reasoned that if we can do away with, instead of diketone, we make it a monoketone, then we will not have that problem instead, right? Now, one more question. There's another subject. Okay. So one more question. What's that famous reaction when you take one mole of aldehyde, a base catalyzed reaction with 1 mole of ketone to give you a reaction? Condensation reaction, right? The beauty of condensation reaction is all your reactants basically they are like addition reaction. They form an atom, and generally it forms a higher molecular compound, and it just kind of in your solvent it settles down, you know, so you can easily filter out. That's the beauty of condensation reaction. It condenses down, you can easily filter out. And your purification becomes much easier. You don't have to run those lengthy columns, right? So anyway, so this is called Claisen-Smith, particularly Claisen-Smith condensation. I will skip those details. You probably try that over that on your Liam reaction examination, but keep that in mind. That's Claisen-Smith condensation, okay? So anyway, so that's how we synthesized. That was the best catalyzed Claisen-Smith condensation was used, and then we, as I told earlier, what we improved upon this was we scraped off one of the keto group and then made it a monoketo group. That way it was more stable. And then we wanted to see whether it has better biodistribution or it dissolves better in water than curcumin. That was another further thing to notice, right? So this is I'm taking you through the pipeline. As I told you earlier, like you do in silico and docking studies, come up with a base framework, and then you do some chemistry on that. And this is what is the SAR studies here. You have different site groups here, site chain structure and activity relationships, right? You have different site chains here, and then depending on this. So we made a series of basically you call it library synthesis. So you have this base structure, and then you basically change only modify these R groups, site chain groups, right? And then see if you change the structure, what happens to the activity. That's what is SAR structure. Okay, that's what a medicine chemist does, or organic chemist, synthetic organic chemist does that. Okay, so that's what we did, and then we built this library of around 30 compounds. And then they were further screened. As I told you, it's a long process, right? So you go step by step. After you synthesize the molecule, you verify it, then you have to characterize it, right? Okay. So will you tell me what are the spectroscopic methods that are there out there which can tell us what exactly? FTI, yeah? Awesome. So you get one chocolate. What else? Okay, you get all collect at the end. Okay. FTIR, okay. So but FTIR, you know, it only tells about your basically functional group, right? FTIR doesn't tell you much about the structure, you illustrate the structure, right? If it's FTIR, it will tell about the functional groups, and so on and so forth. If it is 1H NMR, it will tell you where the protons are located, right? If it is carbon NMR, it will tell you about the basic carbon scale and so that's what is the spectroscopic method. So after we synthesized this library, as I was telling, so next thing we did was analyze this, analyze the structures by spectroscopic methods. As you all know, you already told that. So we did basically FTIR to look at the functional groups. I'm skipping the details, but just giving you the overview. And then NMR, and then finally, final proof of a molecule, actually, if it's small molecule, is XRD. You know, even after NMR, you can illustrate structure. But if you can crystallize it, that's what Rosalind Franklin actually was very crucial in determining structure of DNA. You know. Though Watson and Crick got the Nobel Prize because Rosalind Franklin couldn't live long to see that day, right? To see the light of the day when the Nobel Prize was awarded, and Nobel Prizes cannot be awarded posthumously. Right? So that's why, so Rosalind Franklin she had a very crucial role in crystallizing DNA. You know? So when Watson and Crick, they built the model later, but they arrived at that model looking at the XRD, single crystal structure. So I will skip the details, but basically XRD tells you like when the molecules stack together, when you crystallize it, how it looks in 3 dimensions, and how it in the lattices, how it basically that tells you the exact structure. It's like a proof. It's like a XRD. That's why particularly for drug design, you have to look at the XRD structure of molecule. And there are like complications. Like there are you can add sophistications. You can just take a single crystal X-ray of a molecule, and if it binds to a protein pocket, you can also see how it binds, and then if you can crystallize that entire protein, then you can also look how that looks like, so on and so forth. So XRD is also a very important aspect of screening. Now you made the molecules, you purified them, you confirm that those are the exact molecules by spectroscopic methods and XRD. Now what you do is you do basic screening, whether those molecules have any activity, right? Whatever is your desired activity. It could be anti-inflammatory activity or anti-cancer, depending on that. So I am giving one example because I am continuing on the curcumin analogs here, which I did for my master's. So this is one more, why I am putting this here is all the assays you run, right? The basic chemistry, basic analytical chemistry, the structure, your basic redox reactions, that whatever you run, all these assays are designed on that, whatever analysis you do. The core of all the assays, there is chemistry there in every assay, whether it's viability assay, whether it's a DPPH, in this case it's called DPPH, it's basically, it looks at 2-diphenyl pitryl, something like that, right? But it's generally called DPPH assay, okay, it's very famous. So what it does is basically, so there is a theory of free radical in our body, like radicals are very reactive, right? What are radicals? The basically electrons are not paired there, right? It has a free radical. It's very reactive, right? Our dad asked a son, what do you want to be when you grow up? He said, a free radical. That wasn't reactive. So the radical has another meaning literally, but in chemistry, radical also is reactive, right? Very reactive. So the control. Basically, ascorbic acid that's the vitamin C, that's taken as control here. So a control means when you try to run an assay, right? At the end of it, you have to have something to compare it. If I say I am becoming fat, if somebody fatter than me comes, then I become thin, right? So then I have to have something to compare it, right? That's why whenever you run any bioassay or chemical assay, you always have a control. Yeah, so basically ascorbic acid was a very well-known free radical scavenging vitamin C was put as control. And then compared to that, how these molecules do it, that's how they were screened, right? So as I was telling earlier, hit compounds are, whichever show a little slight problems, those are hit compounds. So the hits were identified after this. So basically, this is not cytotoxicity. You also have to test for mutagenesis. There is one more called teratogenesis. That means if a mother who is bearing a child in her womb, if she takes that medicine, will that affect the going to be born baby, right? So that kind of studies also have to be done. It's toxicity is done in a lot of levels. So basically, that's what James Black says, right? Medicines are used to poison. It's also like, for say, basically, anything can be poisoned, depending on the dose, right? If you take too much of sugar also, that can be also poison. So that's why it's very important. The next step will be to screen viability, cytotoxicity. So I will skip the details, but it's very famous essays for Alamer glue assay. Now, I will go to the chemistry part, I will skip the biology part. Okay, so here comes the biochemistry. As I was telling you earlier, a medicinal chemist has to know all the fields, at least the basics. So you see, like here, there is basically this molecule here, rizogrin, and then you see there is basically inside the body, this reaction happens, and it basically changes the color. Okay, it's the colorimetric assay. So what's the reaction happening here? That's the question for you. And you look at this structure, what's the reaction happening here? What's happening to this molecule here? What's the transformation happening here? Redox reaction. Awesome. So that's what is happening, right? So they say that redox reactions go tandem, right? If one species get oxidized, the other species get reduced, right? So that's what, this is the redox reaction. If you look at the molecular structure of this, you will see like, basically it's a redox, right? Basically, NADH is a coenzyme in our body. It's like it's basically, it's involved in electron transfer processes and metabolism. So that's why it does NADH. So in the live cells, when the NADH can oxidize, basically can reduce, into this colored, like a pink colored species, then you can detect, depending on the intensity of the color change, you can quantify basically how much viability is there, right? That's how this LMR blue assay is designed. So I'm just going through the chemical part of it, right? The chemistry part. So basically viability studies were performed, and then out of those, basically the viability was assessed for those 5 hits, right? So that's how, then we moved further, that they were, also in the viability assay, you see that compared to the non-treated control here, which is like almost more than 90% viability, almost all of these compounds, these hit compounds had viability at least 90%, so that means we can go ahead. If you have a viability of 50%, then that molecule is done there itself, you don't go further. So that's what it was done here. And then basically mechanism of action, why it does whatever it does, right? That's what is the mechanism of action. So basically it's more of a hypothesis, but it's a little bit supported by our assays. Our hypothesis here is basically the curcumin base structure. We can do some SAR there and scrape off its instability and maybe slightly improve its bioavailability, and then it can bind to some of these inflammation related cytokines like LOX, COX, which in the downstream it's in turn like inhibits production of nitric oxide. So these are, again, you see these can be free radical inducible nitric oxide synthesis is one enzyme which basically can generate that kind of so it's also involved in the inflammation. They are also actually, these are also in a way like a radical species, which damage the cells, and that's how the inflammation happens. So basically it can reduce the nitric oxide production, so that means it will show like some basic anti-inflammatory property, right? So that's what is our, that was our summary of this study, So that was my master's work, and then I briefly go through my PhD work. As I was telling, like, as you see, that was mostly like chemistry, right? I was a scientific chemist in my master's, so I was mostly doing chemistry and doing the structural activity and then looking at the trends. But as with my PhD, I lysine is the proteins, the cellular proteins will have the exposed lysine residues, right? So they will have a free amine on them. So if you can modify this amine, what happens if you react NHS with amine? What do you get? Whenever you have to, what is the peptide bond? Amide bond, right? That's a very, very simple step. It's always the NHS chemistry is done, right? NHS to amine, so always it gives you a very robust bond, right? So that's what was done here. So with this free amine, the NHS binding was reacted to get the amide bond, and then that's how the biotin NHS was clicked to the cell surface, And then once you click a biotin is a vitamin which has, in nature, actually biotin is the strongest binding, basically it binds very strongly with avidin, right? Protein. Biotin and avidin is one of the strongest interactions in this. Avidin is found in eggs. So anyway, so that was, this kind of chemistry was done. So in this study, our purpose was when we modify the exterior surface of living cells, we do some chemical. So that's why it's called, from medicinal chemistry it became chemical biology. That's the reason there's subtle difference there. When you are using some chemicals, mild chemicals which are not toxic like vitamins or anything like that. So that's what more or less my study was confirmed. I'll skip the details, but after putting this fluorophores here, we kind of looked at how the fluorophore stays on the cell surface over the time, that kind of experiment done. So the point here is, no matter what chemistry you do, the next step is always you want to try to apply it. Right? This is one of the days of like Hoffman and Woodward, you make like 60-step multi-step synthesis. This is our immune cells. So macrophage is always home to like tumor site, whenever they detect any foreign entity, whether it's a bacteria or an infection or inflammation or cancer, there's this kind of, they have this inherent ability to home to that, right? So we tag the immune cells with green fluorescence, and then for this we use the DAPI coloring for the cancer cells. As you can see, the green cells, how they cells, and then it's called time-lapse microscopy. So there was basically a fluorescent microscope was put there, and at the same place it would take picture every 10 minutes for 24 hours, and then we would stack them all those images and make a movie out of it to see what it does in real time, right? So if you see in the beginning, they were all the cells were random, right? Because we added those cells, they were random, moving all around. Then eventually they start moving towards the cancer cell. As you know, second law of thermodynamics. No system wants to be more, right, entropy, right? Actually, any system wants to go more random. Nothing here. It tries to be more structured. That's against entropy. But why it's happening is because the cancer cells secrete certain cytokines, which the immune cells recognize as danger, and then that's how they move towards it. So keep that in mind. Now you will see the blue colored cancer cells just going round in the beginning, and then the green ones, how they circle them, eventually you see. See that? Right? Eventually all the immune cells are just coming around the cancer, right? So this was the proof of concept I worked in my Ph.D. that immune cells we can use chemical biology tools to modify the live surface of the immune cells, and then they can be, that way we can track cancer in real time. So this is, my engineers are taking it further. So our further plan is to add, if we can attach, just like I showed you in the previous slide, if you have a method to modify the exterior cell surface, right? So you can also probably you do screenings, you do SR studies, and you further come up with hit to lead, and then you go to new chemical entity, and then you do drug entity, right? But this entire thing also is, if you look at the actual drug discovery, this entire lead discovery is only a small part of the entire drug discovery pipeline. So this is only the chemical part. From here also, like this is what the basic research, this is assessment, and this is what the lead discovery. All I was showing you was only this part of it. That's why drug discovery is a long process, and it's $1 billion. A drug could cost more than $1 billion before it comes to a market. That's why it's a very lengthy process. And so I will skip that. So that was I wanted to give you an overview of drug discovery process and what is the role of a chemist there, right? So till now I was telling all this in English. How about in Nepali, right? Questions in the Bible.
क्वेसन दिनु सकिएन है विशेष एकदमै देशलाई माया गर्ने नेपाली माया गर्ने मान्छे है तपाईँहरू उहाँसँग कनेक्ट गर्दै धेरै फाइदा लिन सक्नुहुन्छ मलाई बोलाउनु कारण पनि त्यही हो है उहाँ जुन त्यसले गर्दा उहाँ धेरै धन्यवाद हामी दिनसँग केही पनि छैन कालो चिया होइन अब कालो चाहिँ मैले सबैलाई खानु भने खानु भयो है अनि सरलाई मैले पनि धेरै गर्ने मैले पनि धेरै धन्यवाद दिएँ अब यहाँ 2 बजीदेखि अहिले 15 मिनट ढिलो भइसक्यो सरी उहाँको विशेषज्ञ हामीले त्यसको पुस्तकलाई धन्यवाद सरलाई केमिकल मोडल पनि छ भन्नुभएको थियो त्यो विद्यालय पढाउनु राम्रो किताब छ राम्रो